So my name is Jonathan Duskin. I'm a senior in the neuroscience program, and my presentation is on the development and implementation of a pipeline for determining patterns of volumetric, and you can read the rest. We'll get into what all that means. It's big words that mean a lot of little words that are much easier to understand. All right, so first of all, what is multiple sclerosis? Multiple sclerosis is an inflammatory disease of the central nervous system. So to understand the main signs of multiple sclerosis, picture that I were to throw a baseball at Francesca in the back. So what would happen would be her eyes would send a signal to her brain, and her brain would perceive a ball coming, and she would pick up, and her brain would send a signal to her hand saying, pick up your hand and catch it before it hits you. So in order to do this, you need axons, because axons are the part of the neuron that sends the signal from one neuron to the next. But the other thing is, if she were to pick up her hand after a minute, that wouldn't do her very much good. So you need a signal that travels very quickly, and that's where myelin comes in. Myelin coats the axons and it kind of acts as an insulator and allows the signal to travel very quickly. And I'll show you what that looks like in a second. And then the third is that it causes lesions. This is where the inflammatory component comes in. So what a lesion is you have in your brain, in your head, you have the blood-brain barrier. Keeps things in your blood separate from things in your brain, which is good because when the blood-brain barrier is broken, then white blood cells can travel through your blood, get into your brain, and what white blood cells do all over your body is they cause inflammatory reactions, which in many cases are necessary to cure damage and things like that. But when it's in your brain, this, especially in multiple sclerosis, this sets off neurodegeneration, which is obviously a very big problem. Multiple sclerosis affects 2.1 million people worldwide. There's a wide range of symptoms, as you can imagine. If your body, your neurons innervate pretty much everything that happens in your body. So you can imagine what happens if signals are not being sent or signals are being sent too slowly. There's a wide range of problems. So there's four main courses of the disease. I'm concerned with relapsing remitting, which is this one right here. It's the most common, 70 to 80% of cases. It's characterized by the relapsing phase, which is severe worsening of neurologic function, followed by a recovery or a remission. It's not necessarily a full recovery always, so sometimes you just have a partial recovery back to normal neurologic function. And what happens with relapsing remitting is, unfortunately, it can turn into more secondary progressive pathologies, which are really bad because once it gets to that point, you kind of just fall into a cascade of neurodegeneration. So that's pretty depressing. But the good news is the drugs for MS that are available are fairly good. The issue is that you need to detect multiple sclerosis in the earliest stages of the relapsing remitting phase for them to be useful. Okay, so I don't know what just happened. What is this? Okay, so I'm going to be talking a lot about gray versus white matter. So all gray matter is it's essentially the nerve and cell bodies of dendrites. So this is the gray matter around the outside. These are the dendrites. This is the part of the neuron that receives the signal from another neuron. It comes from the axon of another neuron. And this is the nucleus. And then there's the white matter, which is this area. It's on the inside. As you can see, it looks white. And this is the axons, which is covered by myelin. As I said before, myelin acts as an insulator and allows signals to be sent very quickly. Okay. Whoops. This isn't synced with this. Sorry, this is... Katie, it's not synced together with it for some reason. It's changing it on here, but not on there.
Okay. Let's just try redoing it.
All right, that's good.
Yeah?
Cool, thank you. Okay, so multiple sclerosis is typically characterized as white matter focal lesions. So I showed you what the white matter is. We talked about lesions. It breaks in the blood-brain barrier. And you see this on MRI. So this is usually how it's been diagnosed in the past. But now there's a movement away from that and instead of looking at the lesions, which come and go, instead looking at gray matter atrophy, which is a progressive process that happens over time and which ultimately leads to the neurodegeneration, which is the main issue in multiple sclerosis. So they're suggesting that gray matter changes are able to be seen early on, and also importantly, another study found that it's shown to have it correlates better with disability and cognitive impairment than the focal lesions themselves, suggesting atrophy as a better measure. So in my title, I talk about microstructural measures. So a microstructural measure gets its name because it's talking about processes happening on the nanometer scale. So one of these that I'm very interested in this study is fractional anisotropy. So fractional anisotropy is the measure of the degree to which diffusion of water occurs parallel rather than perpendicular to the neurons. What does that mean? I'll show you. Pretend that this is your axon. So if I were to spray this with a hose or something and cover this, water, because it's intact, water would flow along it. So that's indicative of high fractional anisotropy, and thus it shows that there's not axonal damage. But if I put a hole in this, now if there's a hole in this, some of the water would flow in and go through here, and you would have not only water flowing across, but you'd have it going in many different directions. So that shows lower fractional anisotropy, and it suggests that you have axon damage and demyelination. Another measure that's very, very interesting, and that my PI actually, it's like his champion work, is it's called free water. So free water is describing water molecules that don't exhibit flow and are not bound by their surroundings. What does that mean? It's not important. What's important is what free water measures in the brain. Free water is sensitive to cerebrospinal fluid, which is the fluid that your brain is floating around in your head, and vasogenic edema, which is the really important one we're concerned with. Vasogenic edema, so edema is the pooling of water in tissue. So in this case, we're talking about brain tissue, and I'll talk in a second why that's important. And vasogenic means that it's caused because of a break in the blood-brain barrier, which is consistent with multiple sclerosis. So vasogenic edema results, so after the blood-brain barrier breaks, as I said before, you get white blood cells coming into the brain. The scientific word is leukocytes. And these come into the brain and set off inflammatory reactions. So vasogenic edema is sensitive to neuroinflammation. Free water is sensitive to vasogenic edema. Thus, free water can detect neuroinflammation, which is a very important thing when you're trying to understand about how neuroinflammation leads to neurodegeneration. Okay, so also I said it can either be vasogenic edema or CSF. How do we know when it's talking about CSF, which is not a pathological thing? CSF is necessary. You can see it. All these black areas are CSF. These are the ventricles, and then if you look around the parenchyma, you can see the CSF. So if I'm seeing high free water here, I know it's got to be vasogenic edema because that's not where all the CSF is located. All right, so I created a pipeline, which is basically a set of methods to carry out this study, and the pipeline is fairly general because it can be, I did that on purpose so it can be applied to other diseases, as you'll see later. So what we did was first automated Freesurfer segmentation. I'll show you what that looks like. It segments the brains of, we used 6 relapsing remitting MS patients over 6 time points, segmented their brain into 183 different regions, and then it was able to calculate the volume. Next, what I did was I created a script that could take these segmentations in the brain and calculate diffusion measures. So in this case, it measured fractional anisotropy and free water. So that's how we were able to do that. And then we calculate correlations, k-means clustering. I'll talk about that in a second. So this is what a Freesurfer segmented brain looks like. As you can see, each different area has a different color. And then what you do is you can calculate the volume taken up by a given color. And then you can also calculate the fractional anisotropy in this area. And that arrow is pointing to the axial side, which is what we work mostly in. So main research questions in the study. Are microstructural measures, so that's fractional anisotropy and free water, which measure axonal damage and inflammation respectively, more sensitive than volume changes, which shows atrophy? Hypothesis, yes. Is either microstructural measure, FA, or free water associated with gray matter atrophy? Hypothesis is free water because inflammation leads to neurodegeneration. I mean, that's the hypothesis in multiple sclerosis. So correlations I found, increased white matter free water or inflammation was correlated with decreased white matter volume or increased atrophy in the axons area. So this was interesting. Another interesting finding was increased gray matter inflammation was correlated with decreased white matter volume, which shows that both inflammation in the gray matter and in the white matter caused atrophy in the white matter. Okay, so I have to run through this quick because I'm getting pressed for time. But so we used gray matter, so we used k-means clustering to model the pathology of the disease. So these are 3 clusters. They're made by, so basically there's the script. It tells you the optimal number of clusters. This said 3. So all the gray matter regions in these clusters followed the same general activity given by that cluster. And these model the pathology of MS. So you have decreased inflammation early on, and then a decrease even more and more. So this shows the remission phase of relapsing remitting MS. This shows an increase in inflammation, and it remains high. So this kind of shows that inflammation turning into neurodegeneration. And this one is my favorite because it has the most in it, and it also shows both parts of the relapsing remitting. So you have the initial rise, which is consistent with the relapsing phase, followed by the partial recovery back to the remission phase. And then what I did was I took these same 199 regions, and so in addition to looking at free water changes, I looked at changes in volume and changes in fractional anisotropy. So as you can see, the volume, you can't really tell changes in the volume early on because it's very up and down. So volume is not as sensitive. Whereas fractional anisotropy, it's pretty stable, and then it has a dip at the end. So this suggests that it becomes sensitive towards the fifth time point, which is after about 2 months. And it also suggests that the prolonged inflammation ultimately leads to this decrease. And as I just showed before, you can tell starting very early on that this is modeling the pathology. So it suggests that free water is a very sensitive model. Okay, so then I implemented this pipeline in acute TBI. I got to run through this quick, sorry. And we found 10 distinct regions that displayed statistically significant differences, volume, FA, and free water between acute traumatic brain injury patients and healthy controls. And these were areas associated with vision, visual perception, language, semantic memory processing, auditory, touch, speech production, conscious emotional feelings, motor and sensory pathways and hormone release, and CSF, which suggests atrophy in the surrounding regions. Conclusions: gray matter and white matter neuroinflammation is associated with white matter neurodegeneration. Gray matter degeneration proceeds independent of white matter inflammation because increased white matter free water was not associated with decreased gray matter volume. K-means clustering, as I said before, effective model of dynamics of MS, and inflammation is more apparent in early than later, and I just showed you the TBI results. So future directions: we got to do lesion analysis. I would like to get controls to compare to the MS patients, and I want to implement this pipeline in other diseases. I did MS, I did TBI. It would be cool to do this in chronic traumatic encephalopathy, which is the disease with the NFL players, and in Alzheimer's disease, to gain a better understanding of the relationship between these measures. And that's all. Sorry if I went over a little.